So in this video, I'm going to talk about one of the commonly used methods that we have in plant biotechnology, which is that of Agrobacterium transformation. So this is a method that was super popular in the sort of late 90s, early 2000s. It's starting to be eclipsed a bit by CRISPR, which there's another video about, but it's an important strategy and it's still used, so I want to go through it. So the basis of this method is it's using a biological vector called Agrobacterium tumefaciens, which is a natural plant pathogen. But the way that it infects plants is particularly interesting and involves actually putting new DNA into the plant. And this is why it's used in biotechnology. So I'm going to go through two things first of all. First of all, I'm going to talk about the natural infection system. Because if we understand this first, then it makes it easier to understand what's going on in the biotechnology. So let me just put a line down the middle of the page. Okay, so with the natural infection, what we've got a partnership between two organisms. So one of them is the plant, and Agrobacterium is a soil bacterium. So it's the root that we're thinking about. So there's the root going through there, and then we have our bacterium, which I should draw like this. This is not to scale. This is to show you all of the important bits of the system. Okay, so this is our Agrobacterium cell. Okay, and Agrobacterium is a pathogen. It's quite an opportunistic pathogen, and it infects damaged plant roots. So when a root is damaged, it will release wound signals. So there's a bunch of chemicals that are released, including something called acetosyringone. So there's a bunch of chemical signals that are released from a wounded plant, and they are detected by a receptor at the bacterial membrane. So here's the bacterium. I'm just going to draw in its so that is the DNA genome. Okay, and at its plasma membrane, it has a signalling system made up of two proteins, which are called Vir-A and Vir-G. Okay, so they are a signalling system. So they detect the wound signals, and then they stimulate some events inside this bacterial cell. And what makes Agrobacterium so special is it has a plasmid inside it that is responsible for the infection. So this is a plasmid. So all of the genes that are involved in this process are not in the DNA genome. They're in another piece of DNA called the plasmid, and it's called the TI plasmid. Okay, so we've got an extra bit of DNA inside the cell. That piece of DNA is able to replicate itself. So it has, one of the things in it is is what we call ORI. So ORI equals origin of replication. So that's a particular sequence in the plasmid that means that DNA polymerases can recognise it, so we can make new copies of the plasmid. So you have to have an ORI in there in order for the plasmid to be able to replicate. Sorry. There's then two important different bits of this plasmid. So one is a bunch of genes which are called the Vir genes. So Vir equals virulence. Okay, so what happens is that Vir-A and Vir-G detect these wound signals, and then they stimulate expression of those Vir genes, so you get a bunch of Vir proteins. So you've got a variety of different Vir proteins. And these have a variety of different functions, which are all to do with a particular piece of DNA that's on the other side of the plasmid. So we've got all the Vir genes on one side, and on the other side of the plasmid, we have a piece of DNA which is called the tDNA. Okay, and it's defined by having a left border and a right border. So that is 25 base pairs of sequence, which is basically a recognition site. So there's one at one end and one at the other end of the tDNA. So that's sort of marks it out as this is a special piece of DNA that the bacterium should do something differently with. So what we have, one of those things that those Vir proteins do is to make a copy of the tDNA. So it's a single-stranded DNA copy of the tDNA. So we make a copy of that, and then we package it in these proteins. So some of the Vir proteins are to do with copying, some of them are to do with packaging. Okay, so those are Vir proteins that are packaging it. And then other Vir proteins create what's called a type 4 secretion system, which is a pilus. So what that means is there's a bunch of proteins that make a connection between the bacterial cell and the plant cell. So we've now got a direct cytoplasmic connection between the two organisms, and it's called a pilus. So we've made a copy of the tDNA, we've packaged it up in all of these Vir proteins, we've made a secretion system, so that tDNA can now go into the plant, and it can insert into the plant genome. So if I just divide this up as a cell here, and then here we've got the nucleus. So this is the cytosol, and we've got a bunch of DNA. Let's just try to show it like that. So this is the plant genome. So the tDNA, again catalysed by more Vir proteins, gets inserted into the plant genome. So this is was made from a bacterial plasmid, it's been packaged up, it's been transported across, and it's now been inserted into the plant genome. And now the plant just recognises it as a piece of DNA. It doesn't think it's foreign. It's able to use that DNA. The tDNA contains genes for making cytokinins and auxins. So those are plant hormones that cause unregulated cell division. So Agrobacterium infections, you end up with a tumour forming, so you get cell division. And we have the genes for opene synthesis. So openes are nitrogen-rich compounds. So what happens is the plant starts making the genes that are encoded on the tDNA. So we get a tumour forming, and we get openes are made, and those are food for the Agrobacterium. Okay, so the Agrobacterium never gets inside the plant cell at all. It does all of this remotely, but it makes a copy of its tDNA, puts that tDNA into the plant starts using the tDNA, it doesn't realise that it's foreign genetic material, so it starts to make a tumour, so there's lots and lots of cells, and those cells are all making opines, which are food for the Agrobacterium, so they get chucked out of the plant, and the Agrobacterium can just eat them in the soil. So the Agrobacterium is an incredibly efficient pathogen. It's not having to do very much, it's engineered the plant to do its dirty work for it and to make it a novel food source. So that's the natural infection system. So let's think about the engineered system and how we might manipulate that in order to do some biotechnology. Okay, because obviously if we've got an ability to stick bits of DNA into plant genomes that plant genomes can read, that's a really, really useful tool for us, but we don't really want to be making tumours and to making these opines. So we need to manipulate the system a little bit so that we can put what we want into the tDNA rather than doing what the bacteria wants. Okay, so let's, I'm just going to draw out the Agrobacterium cell again. I'm not going to draw the root out. We actually transform via flowers in Arabidopsis certainly, or via plant tissue culture. So I'm not going to show the plant bit, I'm just going to show the Agrobacterium bit. Okay, so again, there's the DNA genome, which is pretty irrelevant in this system. It's obviously needed for the Agrobacterium to grow, but we don't need to worry about it too much. We keep the Vir-A and Vir-G. That works quite nicely, but we might have a synthetic signal that we use to activate that signalling cascade. But the signalling cascade was useful, okay? And what we've done to engineer it, so on this one we had one very big plasmid in the Agrobacterium. And very big plasmids are difficult to work with in the lab. What's better is to use smaller plasmids that you can put in and out of E. coli and sort of chop up and do whatever you like with. So one of the things that we've done in the engineered system is to split this big plasmid into two plasmids. So both plasmids will need an ORI, otherwise they won't be able to get replicated. Okay, but we've now got two plasmids, so we've got one that we still call the TI plasmid, and we've one that we now call the helper plasmid. So having two small plasmids is easier to work with than one big plasmid. Okay. We still want all Vir genes. They are a useful thing because they did all the hard work of cutting it out, of packaging, of making the secretion system. We want those all to work exactly as the way they did before to make the Vir protein. So we keep those genes, those are useful genes for us. So we keep those on the helper plasmid. But what we also have on the helper plasmid is a selectable marker gene. So we put in a selectable marker. So that might be something, it's usually an antibiotic resistance gene. So what you do is you have your antibiotic resistance gene, and it does have a prokaryotic promoter. So this means, so it's a prokaryotic promoter, so it can be switched on by a bacterium. And if it encodes an antibiotic resistance gene, that means that it's very easy to tell which bacteria have the plasmid in it correctly or not. So if you grow a bunch of cells in the presence of an antibiotic, so let's say it's kanamycin, okay, only the cells that contain this helper plasmid will be able to survive because it's got the kanamycin resistance gene in it. So normal cells would be killed off, but if our bacterium has got the helper plasmid with an antibiotic resistance gene, then they will survive. So we can guarantee by using the selectable marker that they have got the Vir genes. Okay, so that's the helper plasmid. But we also have this second TI plasmid. So if we're going to have a second plasmid, we're going to need a second selectable marker, but a different marker. So it's another selectable marker. Again, it's got a prokaryotic promoter, so it can be switched on in bacteria. But it's going to be a different antibiotic. So if this one was kanamycin, this one might be neomycin, for example. Okay, so we now actually grow our bacteria with two different antibiotics to check that they've got both of the plasmids. Okay, so you can grow bacteria with multiple different antibiotics, and if they're resistant to both of them, then we know that they must have both the helper plasmid and the TI plasmid. So what's left on the TI plasmid? Well, on the TI plasmid, we will have our tDNA, which is still has the left and the right borders. Okay, because those were the recognition sites. That was what the Vir genes or the Vir proteins were going to bind to make a copy of it. So we need those to stay in there. But we want to change what's in the tDNA. So in the original tDNA, there were cytokine and auxin genes and opium synthesis genes. They're no use to us for biotechnology. What we'd like to do is to replace what's in that tDNA with something that's useful to us as scientists. So I'm just going to draw the tDNA out. So we've got the left border and the right border. So that defines the beginning and end of the tDNA. So we will put in our gene of interest. So that could be anything we like. Okay, so that might be green fluorescent protein, it could be something from another plant, it could be a gene from a bacterium, it could be anything we like. We can insert into the plasmid using standard plasmid engineering techniques. We'll want that gene to be switched on in a plant. So we'll need to make sure that we've got a promoter. So I'll just put it on here just as a reminder. So the open reading frame makes the protein in a gene. The promoter, so the arrow that I show at the front, is the switch or the control region. So when you're designing a system, you need to think about both what is the product that we're going to make, so that might be green fluorescent protein, but we also need to think about the switch, and this is going to be made, it's going to be expressed in the plant. So it does exactly the same thing as it did here. It's going to be inserted into the plant genome and then used as just an extra bit of DNA. So it's going to be expressed in the plant, so it needs a plant or a eukaryotic promoter, a eukaryotic control region. So we've got our gene of interest, but what we can also do is to put in yet another selectable marker. Okay, so we've got marker 3 goes in there with a eukaryotic promoter. Okay, so what this is for, so it's another selectable marker, but this time when the tDNA goes into the plant genome, we need to be able to tell which plant cells have got the tDNA and which ones haven't. And we can do that in exactly the same way. So we can have that as either an antibiotic resistance or a herbicide resistant gene. We can use both if we're in plants. So if we have that selectable marker, we can then, when this thing goes into the plant genome, we can then select for which plant cells are resistant to whichever antibiotic or herbicide that we make it resistant to, so we can tell quickly and easily which ones are going to have our gene of interest, because if they've got the gene of interest, they should also have the selectable marker. Okay, so that's our tDNA in the engineered system. So we've taken out cytokine and auxin, we've taken out OP in synthesis, we've put in our gene of interest, so whatever we're actually trying to modify with, and we've put in a selectable marker into the tDNA so that we can tell which plant cells have been transformed and which haven't. Okay, so we've taken this natural infection system, we've kept the bits that work, we kept Vir A and Vir G, we kept the Vir genes, we kept the origin of replication, we've put in some selectable markers to make our lives easier so we can see which cells have got the plasmids, which ones have got the constructs, and then we've replaced it, the OP genes with our gene of interest that we can then stick into the plant genome. So that is how Agrobacterium transformation works, and this, as I say, was a really, really powerful technique. It's been used loads and loads and loads. It's now been taken over somewhat by CRISPR-Cas systems, and there'll be another video on that sort of system, but Agrobacterium has been the absolute workhorse of plant transformation for a while and is still very useful when it comes to plant biotechnology.